Morning, students. In previous lecture, we discussed about the structural protein of muscle, the actin and the myosin. Actin and myosins cover the maximum part of the muscle proteins, but the other proteins like regulatory proteins and the structural proteins are also very important in muscle contraction. Today, we would discuss about regulatory proteins and structural proteins found in muscle structure or in a sarcomere of muscle. I am Dr. Ashok Kumar Siroya, Department of Zoology, SPC Government College, Ajmer. Now, start with regulatory protein. The regulatory proteins regulate the muscle contracts. Muscle contraction is controlled or regulated by these proteins. The first one is tropomyosin and the second one is troponin. If we see the tropomyosin, it has the molecular weight of 64,000 units, and the one molecule of tropomyosin is attached with 7 units of actin proteins. Or we can say that one molecule of tropomyosins cover the 7 globular units of actin protein. And the second one is troponin. Its molecular weight is from 70,000 to 40,000 units. These are of 3 types and associated with the tropomyosin. So the tropomyosin is attached with actin and the troponin is attached with the tropomyosin. Tropomyosin is a rod shape about 41 mm long, consists of 2 unique unidentical or alpha and beta chains coiled around each other. These are 2 forms of tropomyosins. Tropomyosin A, or it is also known as paramyosin, and tropomyosin B. Tropomyosin plays an important role in sensitizing the contractile proteins to calcium. Means it conveys the message to the endoplasmic reticulum, and the calcium is responsible for muscle contraction is released. Troponin, the second one, troponin are made up of 3 types of components. These are troponin C, troponin I, and third one is troponin D. Troponin C binds with calcium ions. Troponin I functions as initiator of making bondage between myosin and actin. And the troponin T is bind with tropomyosin. As we discussed earlier, that troponin is attached with tropomyosin and tropomyosin is attached with actin. So if troponin pull the tropomyosins uncover the actin active site and make the site available for myosin head. The third one, or the third type of proteins are structural protein. The structural protein are of three types: alpha actinin, C proteins, and M line proteins. Alpha actinin has the molecular weight of 95,000 units and found on Z line with rod shaped structure. So the alpha actinin form the Z line. And second, C protein, it is found in between the myosin filament on A band. It is the initiator to bind with calcium ion and initiate the muscle contraction. Third one is M line protein. It is found on the middle part of H zone and make M line on it. Its main function is to maintain myosin filament on its own place. All proteins which we have discussed are exhibit in this diagram. Here, the fibrillar actin, the myosin, and on the myosin, these are the myosin head, and the tail part become a rod shape here, and the actin has all three: tropomyosin, troponin on the fibrillar part or globular part. Here the tropomyosin covers the active sites of actin and troponin bind with tropomyosin. These are the structural protein on Z line, on M line, and here on A band. Here, area between Z line, two Z lines, we can say it is sarcomere. And the sarcomere is a unit of muscle fiber. Area around the H zone known as A band, which is darker, and area around the Z line known as I band, which is the lighter area. Between myosin and the Z line, there is another structural protein known as titin. It is elastic filament. In next lecture, we will discuss about mechanism of muscle contraction.